Hello and welcome to Face with Dr. Bruce Jarrell. I'm Alex Lakowski, Executive Director of Media Relations. Today, Dr. Jarrell will talk with two infectious disease experts from the School of Medicine's Center for Vaccine Development and Global Health, Drs. James Campbell and Wilbur Chen. Before we begin, let me say that this program is being recorded and will be posted on our COVID-19 website, umaryland.edu/coronavirus. For best viewing, please select the grid view so that you can see all of our panelists as they talk. A little later in the program, there will be an opportunity for members of the audience to ask questions. Please post your questions in the Q&A section on your screen. And when the time comes, we may call on you to ask your question, so please listen for your name and speak up. Now here's our host, UMB Interim President, Dr. Bruce Jarrell.
Good afternoon. It's a pleasure to be here, and I'd like to welcome all of you to this seminar. Our plan is to have a series of seminars over the next several weeks to provide you with information, knowledge, other types of pieces of information for you to think about the COVID epidemic, the pandemic, and ways to think about it and know what's in store for the future. As you may know, the University of Maryland, Baltimore, and particularly the School of Medicine, has a very long history in terms of infectious disease research. Its Center for Vaccine Development is well into its 50 years and has made many great contributions to vaccine and infectious disease control. Our Institute for Human Virology is also extremely well known around the world for its research in HIV and AIDS. In addition, we have a very distinguished faculty in the School of Medicine and the other schools, some of which do infectious disease research. The Department of Microbiology and Immunology is a great example of that, where there is deep expertise in coronavirus research as well. This afternoon, we'd like to focus on vaccines, where they come from, how we develop them, how we test them, and then what happens to them after they get approved, how effective they are, etc. We have the great fortune of having two vaccine experts with us today. The first is Dr. Wilbur Chen, who's an associate professor in the School of Medicine and a member of the Center for Vaccine Development. Dr. Chen is an adult infectious disease expert and serves as chief of the Adult Clinical Studies Program in the Center for Vaccine Development. He also serves on the governor's task force for the COVID pandemic. Our second panelist is Dr. Jim Campbell, who's a professor, again, in the Department of Pediatrics this time, but with the Center for Vaccine Development, also an expert on vaccines, and serves as chief of the Pediatric Clinical Trials Program in CVD. He's also senior vice chair of our Institutional Review Board, and so is very experienced on the proper conduct of human clinical trials. So I'd like to welcome them and basically begin with having each of them give their view on vaccine development over the years and some recent vaccines that we've had developed that the University of Maryland School of Medicine and CVD has played a key role in. Maybe I'll start with Dr. Chen. Dr. Chen?
Sure. So I started my career at the Center for Vaccine Development back in 2006, and back then one of the emerging infectious diseases that we had to address was avian influenza, H5N1, if many of you might have remembered that. We also performed studies for the 2009 H1N1 pandemic influenza. And then more recently, our center was involved with Ebola and Zika virus vaccines. So over the many years, we've been involved in a lot of pandemic and emerging infections. And this coronavirus represents the latest one, but of course it's been a great threat in a pandemic. So here at the Center for Vaccine Development, we've been doing vaccine trials for many years, and I think that, again, we're in a position to try to respond as best as we can collectively as a group effort, along with the national responses that we've seen nationwide.
Great. Thank you, Dr. Chen. Dr. Campbell?
Yeah, thanks for having me, Dr. Jarrell, and thanks everyone for listening in. As Dr. Chen just said, we've been involved in lots of vaccines. Essentially, if there's a vaccine that is licensed, the CVD had a hand in some part of the development of that vaccine, and in most of the experimental upcoming vaccines as well. We, as you said, Dr. Jarrell, I run the maternal child section, so the pediatric vaccine trial section, and we recently reviewed the studies that have been done in pediatrics well before I arrived, but I was looking through the pediatric schedule, the entire schedule from birth to 18, and every single vaccine on the schedule at some point and in some way was tested at the CVD and at our sites. So we have a number of pediatric clinical sites. So whenever there's an emergency like this, as Dr. Chen said, we're also, we often have to drop other things that we're doing or slow them down for a while so that we can get involved, like Ebola, and Zika, and now.
So Dr. Campbell, tell us how vaccines work.
Yeah, so I think a good way for the person who maybe is not in the medical or the immunology or vaccinology world to think about vaccines is that we over time have figured out what is it about the human body and the immune system that makes a response that after being infected, you then can be protected from that infection. And a vaccine is a way of essentially tricking the human immune system into thinking that it was previously infected and then developing a response, meaning immune cells and immune proteins that last for a long time. And when you then are exposed to whatever that infection is, be it virus or bacteria or parasite, that then your body responds in a way that protects you. So it's through various means of probing or hitting the immune system and then getting it to respond in a way that in the future you would protect it. So we try, often it's a way through thinking about the natural immune responses and then trying to bolster through vaccines to get us to be protected against individual infections.
And are vaccines just for viruses, or do they apply to other organisms or compounds?
Yeah, I mean, I'll take that, Wilbur, and then you take the next one. So, yeah, it's not just viruses. So for infections, we think, we divide them up into different types of organisms or germs. So bacteria, viruses, parasites, and fungi. So it's primarily the vaccines that we have available to us today are against viruses and against bacteria, but people are also in experimental ways looking at parasites and fungi as well. But yeah, it's all of those different types of organisms, and it's different ways of trying to attack them because they tend to infect you in different.
And how long when you get a vaccination, how long does the effect last for? A lifetime or shorter times or booster shots? What's the thinking on that? Dr. Campbell?
Yeah, I'd be glad to take that. So it depends on the germ, and it depends on the technique that we use. So you can kind of think of vaccines as multiple different, what we might call platforms. So there's different approaches developing them, and some of them are either we call them inactivated, but some people would call them like parts of the germ or killed or dead germs. And then there's what we call live or live attenuated. Attenuated just means weakened. So some of the vaccines that we have, like measles, mumps, rubella, chicken pox, yellow fever, these are living organisms, but much weakened compared to the thing that would normally infect you. Those tend to give you longer-term protection because they act similar to natural viruses and bacteria, and so you tend to be covered for life or nearly covered for life with those. Whereas the subunit or the killed or inactivated types, those we often have to give multiple immunizations as what we call a primary series, and then at some interval afterwards, give booster vaccinations to get your immune system back up to being protected. I mean, that's a generalization, but that's typically the way that it works. Things that are alive and weakened give you much longer protection, and those that are killed or subunit, as we call them, little particles, give you a little shorter protection.
Thank you. So that's what my booster shot was for.
That's why you're getting your every 10-year tetanus shot.
Including the ones I didn't get, I presume. Yes.
Right.
So Dr. Chen, what's been the effectiveness of these vaccines? Have they eliminated diseases from the face of the earth?
So there are some vaccines that have been very successful, like smallpox vaccination, which has essentially eliminated smallpox. Yellow fever is another very effective vaccine that we'd like to see get deployed throughout more of the world into other spaces that are difficult to get to. So these are developing country settings, very rural areas that we would love to have it deployed in a better way. That is another very effective vaccine, but we have not eliminated. Measles is something that was on the way of elimination, but then over the past couple of years, kind of a reverse trend where we're seeing localized outbreaks in communities or populations where there's not very much vaccination. But the bottom line to your answer is that there are some vaccines that are so highly effective that we start thinking about elimination. And then there are other vaccines that are just not as good. Influenza comes to mind. That's the one that you have to get every year, update the vaccine. We're hoping that we're going to, through more research, get to better vaccines for influenza, and maybe eventually get something called a universal vaccine, which would be maybe longer lasting and maybe protect you against the next pandemic.
So in influenza, we get a shot each year, and you make a best guess at what the strain of the virus would be. So how does the virus go about changing from year to year? Why does it do that? How does it know to do that? Or what are the circumstances under which it just changes?
Yeah. So the virus is not smart necessarily. It's just nature that as it goes from person to person, that infection, that virus actually has what's called low fidelity copying itself. So it may drift over time, and that means that basically the genomic sequence of it may change over time as it goes through a population. So that from year to year, you may see slightly different looking viruses just because of the gene changes that happen from copy to copy of the virus, because it's so many different people in a population. So oftentimes we're trying to predict what is the virus exactly going to look like so that we can get a vaccine against it. Sometimes we select wrong. Sometimes we select well. It's called a good match or a mismatch for good or bad use. Select those viruses.
So, yeah, if I could interject, I think we in the infectious disease world and vaccine world, we tend to anthropomorphize our enemies. So as Wilbur said, it's really just chance mutation that eventually leads to viruses that rift in a way that allow them to escape our ability to, to be protected. But we tend to think of them as, smart or wily, if you will, like this current coronavirus that we're dealing with. How can a virus kind of figure out how to move person to person, kill enough people that it scares everyone, but at the same time not kill so many people that it can't continue in its lifespan to move on and on. And I think knowing your enemy is sometimes the way that we can try to attack how to get back at the enemy and try to prevent it from continuing its spread.
So the first vaccine for smallpox was discovered sort of serendipitously by a very astute observer. How has the identification or the development of vaccines changed over the centuries? And link that up to today. How are we going to develop the vaccine for this particular virus?
Yeah, I can start with that one, and then Wilbur, you want to jump in after. So if you want to go back in that history, so Edward Jenner is the gentleman you're talking about, so English country physician who noted that milkmaids who developed cowpox, so they got little blisters on their hands from milking cows, and it was because they were getting a cow infection essentially, but caused a very minor infection to them, just little blisters on their hands, that they didn't seem to get smallpox, which was a horribly devastating human infection. And so Jenner thought, well, what if I purposefully infect people with cowpox? Maybe I can prevent smallpox. And that is the beginning of vaccinology, essentially. The word vaccine even comes from vaccin. Vaccinated, it means vaccin is cow. So it comes from vaccinia or cowpox. So that's really that's the history of vaccination. What's happened over time, and maybe I'll let Wilbur, after I give you sort of what's happened, he can tell you what the current approaches are to COVID. Is we've obviously in all aspects of medicine we've just become more and more modern. So we've been able to go from just those guesswork where they didn't even know what a virus was really, to knowing all the way through how viruses work and bacteria work, how our immune system works. We also know what genetic sequences there are now. And for this virus, as many people probably saw in the news, that from the day it was discovered until we knew the entire genetic sequence, a very short period of time, weeks. And so we've been able to go from empiric science, meaning let's guess what might work, to more directed genetic or other means of attacking vaccine approaches. So now we have at our disposal lots of different approaches and platforms that we can try to use to try to figure out, well, for this virus, which would be the best to protect us. So I don't know, Dr. Chen, Wilbur, if you want to speak about what some of the options might be for COVID.
Right. I think just building upon what you've already said, which is that as we learn more about science and microbiology, learn more about how to make better vaccines. So, when we started being able to culture viruses, then we started being able to manipulate those viruses. When we started to learn how to grow bacteria, then we could manipulate those bacteria, purify them, figure out, the cell surface of this bacteria actually has components in it that are protective. So then now we can make more purified vaccines. Then we started going to the gene age, when we started understanding genomics. Now we could actually sequence these bacteria or viruses, and now we're at the age where just within a few days, not weeks, that we can sequence a pathogen and be able to understand about that pathogen. So what we have now are vaccines that use platforms where you can basically take a gene sequence, so you don't even need to be able to grow the bacteria or the virus, and you can basically replicate a vaccine that will fight against that particular pathogen just by knowing the gene sequence. So we've come a long way, even in just the last 20 years, from the standpoint of making vaccines. So I think coronavirus right now, what you're seeing right now is that the first sets of vaccines that are being developed and deployed under testing are these RNA or DNA vaccines. The Moderna one that you hear about from that's the NIH vaccine, the Moderna vaccine, is basically an RNA-based vaccine, again, using the gene sequence that we know about this virus. And there are other ones that are based on the DNA sequence. So again, it's really exciting because we can now develop these vaccines upon faster recognition of these pathogens.
Now, so we've been able to use modern techniques. We certainly had modern techniques with Ebola and Zika, and we've certainly used modern techniques for the AIDS virus, attempting to get a vaccine there. So in the Ebola, how rapidly was that, or Zika for that matter, how rapidly was that vaccine developed, and how effective has it been now that it's been out in people?
Go ahead, Wilbur. You can go ahead.
No, I was going to say you could go first, but I can go first. So for Ebola and Zika, the great advantage was that already had platform technologies that had been tested and evaluated for both of those pathogens. And so again, it was an instance where we could sequence the genome of the pathogen of those viruses and be able to plug that right into a platform and be able to, in the case of Ebola, express it in a virus, in a different virus. One of the instances is something called ESV, or vesicular stomatitis virus. There's another vaccine that's made from a modified Vaccinia ankara, which is actually a smallpox vaccine, but they basically took that weakened virus and plugged in the gene sequence for the Ebola protein, glycoprotein, which is the protective part. So all of those basically were tested very quickly in clinical trials, and the Merck vaccine is the one that was licensed just recently. So that actually took many years to develop the platform, but once you have the platform, you use it broadly for other types of viruses. So Zika is another example where they took that existing platform and plugged in the gene sequence, and there you go. We had a vaccine that could test pretty quickly. And that's also the instance here for coronavirus.
When you say pretty quickly for Zika, what do you mean by that?
Well, the traditional sense of vaccine development is that it would take at minimum of 10 years. And the fact that we could recognize the Zika virus in South America emerging, and within less than 6 months have a vaccine in clinical trials, was something that at that time we thought was a great accomplishment. Now, with the coronavirus, we were able to sequence it in, I think, the end of January, and go into clinical trials at the end of March. Again, just really surprising how fast we can go into clinical trials these days. Now, that's probably the fastest that we can go, because once you have the recognition of a pathogen, you still have to do laboratory testing and some animal testing, so what we call animal toxicology testing, before you can go into human trials. So I don't think that we can expect that we're going to be even faster within days, but just the fact that we were able to truncate it to just 2 months is a great accomplishment.
I think, one thing that people sometimes don't think about in terms of these rapid development is all the pieces that need to come together simultaneously. And I think that's one of the reasons, for example, with the Ebola vaccine, that they moved so quickly, is because leadership brought together all of the pieces that typically are kind of more working in series and had them working in parallel. That is, you need the regulatory body, people who make the decisions on go or no go, to be on board. And for example, for the Ebola vaccine, we had IRBs all over the world, WHO in Mali, in Guinea, at University of Maryland, Baltimore, everywhere, all having to kind of work together. We can't wait for one IRB to finish before the next one starts. WHO has to be involved. The manufacturers have to switch from whatever they're currently manufacturing for their routine vaccine. Say, okay, we'll take this plant now and we will make the vaccine that is under development for the new problem. The clinical trialsists, the people who need to write the protocols and run all of the studies, need to be on board quickly. The funding, it can't be done for free. Nothing's done for free. So where you get that money from, who puts the money in, how you get all the contracts in place, all of those things happen for Ebola at a speed that I had never seen before. I think that no one, including even the people that have been around way longer than Dr. Chen and I had ever seen before. It's that kind of coordination that really can lead to things moving more quickly. One of the problems that's occurred now with why it's taking a little bit longer for coronavirus vaccines to get off the ground, even though we are moving quickly, is we're having to start kind of at the beginning. We don't have any platform of a coronavirus that's gone far enough along for us to be able to just, as Dr. Chen said, plug it in. And that one of the reasons for that is that SARS and MERS, people out there may have heard of the other, newer coronaviruses that came along. SARS in 2002, and MERS 10 years later. When those came out, everyone was very worried about them. Lots of vaccine activity went on. And then without having a vaccine, they kind of did not cause as big a problem as we thought. And all of that energy kind of went away. And those vaccines did not get far enough along in development for us to have them sitting out there ready to go where we could just plug in the new SARS-2 platform. So that's what we're hoping does not happen this time, that once we go further along in this vaccine development, that one, we will have a vaccine for this particular coronavirus, but that we will have multiple platforms available that we can just plug in when whatever new one comes along.
So when you've got a vaccine candidate, why do you have to go through all this trouble of testing it in cells, testing it in animals, and then these human trials? What's the big deal? People are dying from coronavirus. Why don't you speed that up? Why not?
Yeah, I'll start with that, and then we'll let Dr. Chen go. So first is we in the world of vaccines tend to hold our safety bar, I think, as high as anyone does for any biomedical intervention. And why is that? Because most vaccines, once they're approved, get deployed for the entire population. And the amount of safety that you have when you are giving something to everyone in the population just goes up higher and higher. We have, 350 million people in the United States, almost 4 million children born every year in the United States. When we say, okay, this vaccine is now safe and ready to go, and every single child is going to be getting it, 4 million, only some of them are going to be exposed to whatever the pathogen is. So you need to have real high bar for safety. And if you're talking about a biomedical intervention when someone is sick, the risk-benefit ratio is a little bit different than when you're giving it pre-exposure, and many of those people, all those people, are not sick yet, and so you need to make sure that it's completely safe. So we do like to make sure when we have things like this where there's an emergency that we make things happen as fast as possible but still not cut any corners. Because if we were to say, oh, just get that vaccine out there because, yes, you have a vaccine, so just give it to everyone. And then we're wrong. I mean, there's two big what-ifs if we're wrong. But one what-if is, what if the vaccine does not protect you, it actually makes the disease worse when you're exposed to it? That would be the worst nightmare for people in the vaccine world and for everyone else who thinks the vaccine is potentially going to protect them. We need to absolutely be sure that it is not going to make it worse than if you never got the vaccine. And then the second what if is, what if you start putting your money into one thing that looks like the best approach, and then you're ignoring the other ones that might be also better or good approaches? So I think that's why we're trying to push to make sure that lots of different approaches are looked at early on. Then as ones kind of rise to the top, that we start developing those further. So we are we want to make sure, obviously, that it is immunogenic, meaning it stimulates the immune system. It is going to be efficacious, is going to protect you against the disease, but of course that it is extremely safe. And when we say safe, we're not as worried. Most people are thinking about sore arm or feeling a little bit tired or something. That's most of what we're worried about with safety of vaccines, very minor things. Yes, those are very important, but then there are potentially major safety concerns. And we don't want to skip over making sure that we don't, that those are not a problem with any new vaccine.
Well, what kind of major problems?
Okay, so there have been vaccines in the past that have, well, they're not on the market, obviously, because they've shown to be a failure. But if we go back to the 1960s, there was a vaccine for another relatively severe, not this severe, but relatively severe respiratory virus called respiratory syncytial virus, that in most of adults like us causes a cold. But in little children and immunocompromised people, it can cause very severe pneumonia and death. There was a vaccine back then that when given to children, rather than protect them from RSV, if they were exposed to natural RSV after getting the vaccine, they had worse outcomes, more likely to be hospitalized, more likely to go to the intensive care unit, and even a few children that died. So that's the kind of, we don't think that the current vaccines for coronavirus that we're developing are going to cause that, but we need to feel really confident that that's not the case. We know now through modern science why the immune system reacted the wrong way to those old viruses, and we're never going to go down that path again. But we want to make sure with every new respiratory viral vaccine that we develop that that's not a possibility, and that we prove that's not going to happen. Because if we're doing it in 10 people or 100 people as part of a clinical trial, we can study those things really carefully. Once someone says, now just go out there, why not use it for everyone? It's because once you give it to 1 million people and it's already in their arms, they get exposed, you can't take it back. So we don't want to get to the point where we've deployed vaccines that are unsafe. We want to know that in clinical trials. When we feel confident, then they get deployed.
Dr. Chen, anything to add to that?
I think that was a great discussion. I think the bottom line is vaccines, we want them to be safe. We also want them to be vaccines that the public will have high confidence in, because the public perception, even if a vaccine is safe and the public does not perceive it to be safe for whatever reason, then there won't be good uptake of that vaccine. So in the setting of a pandemic, I think it's easy to tell people that there's a vaccine and there will be high acceptance. But when you have other vaccines like influenza every year, there are large segments of the population that say, oh, that's not an important vaccine. Or even in measles, we've seen that in certain segments of the population, confidence in the vaccine has diminished, and that's resulted in the resurgence of measles. Measles vaccine is not any different in its safety. It's actually one of the safest vaccines ever developed because we've given it to so many people. But for whatever reason, confidence has dropped. That's part of the vaccine safety question. It's the question that people interpret it as perhaps a concern. So again, going back to your question, these are the things that we think about for vaccine safety. We want to make sure that as we go through the testing, that we ensure that these vaccines are safe.
So I've had the pleasure of having both Dr. Chen and Dr. Campbell for my doctor because I've participated in CVD clinical trials. I guess with Dr. Campbell for H5N1 and Wilbur with you, swine flu, was it? Swine flu?
H1N1 in 2009?
Yes, right. Yes. They wanted people in my age group, and I was one of the younger age groups, so I volunteered. In any event, what is the age effect of vaccines, and why did you want people in older versus younger versus real young groups?
Want me to take this, Jim?
Yeah, please.
Okay. So, for adults, we know that for whatever reason, older adults in general don't respond as well to vaccination as young adults. And usually it's an age effect where you see people in their 70s or 80s compared to people who are in their 20s or 30s for adults. It could also be looked at if you look at the immunocompromised versus those who have normal immune systems. So there is an age effect. And so what you see, at least for influenza, for shingles, you see vaccines that are specifically designed for older adults. And that's because these types of infections actually occur more commonly or have more severity in older age. There are other vaccines, of course, that we have to titrate and make a little bit different for children as compared to adults. And so Jim can talk about that. But there are some infections, again, that are more common as we age and get older rather than when we're young. And you can think about urinary tract infections, tuberculosis, which usually has a resurgence more commonly in older adults, also compromised as well. So that's why we have sometimes different vaccines for different age populations.
Right. So one other question before we get to people's questions. So where do you think we stand on the development of the vaccine now? for the COVID-19 virus. What's your sort of forecast and prediction? When do I enter your clinical trial?
Maybe I'll start, and then Dr. Chen, you hop in. So the good news is there are a huge number. Essentially, everyone who's involved in the world of vaccines is somehow involved in the work of COVID-19 vaccines. So my last read is that there are more than 50 academic centers and manufacturers who have a product that is somewhere along the stage of development. Now, most of those are not yet in clinical trials. I believe that there are 4 clinical trials. For those not in this world, means people trials. So when the vaccine is in a person, we call those clinical trials. Some of them are still in preclinical, so meaning in the test tube or in animals. But a number of them now have gone into what we call phase 1, the first clinical trial. And the idea is to move them as quickly as possible, but still feel safe about them from phase 1 to 2 to 3. So, I mean, that is the good news, that there are, across every type of vaccine that we know, essentially, so what we call vectored vaccines. So those are things that we talked about before, where you take a different organism that you already know how it works and how the other vaccines with that organism, et cetera. And then you take the gene from COVID-19, SARS-CoV-2, that virus, stick it into the vector, and have that vector express on its surface the gene from the SARS-CoV-2, the new virus. So you can use like an old vaccine and then modify it slightly so that it can protect you against COVID-19. Or we have ones for mRNA and DNA. There are ones that are live attenuated. There are subunits, what we call recombinant. So you just pump out a protein, essentially all the different types of vaccines. But the bad news, as I said before, is that they're not far enough along that we're going to be able to do an efficacy trial right away. So we are going to need to go through all of these phases over the next year or year and a half for us to be able to get to the point where we've proven that the vaccine, one or any, or
Dr. Chen? I'm sorry.
No, I'm done. Go ahead.
Okay. Jim, I think you said it. The question was basically about the status of vaccines. So we have a number that are in early phase. We have even more that are getting ready for the early phase human clinical trials. We don't have anything in the mid stage or the late phase, which is the bad news. As Jim was kind of talking about, we have different platforms, but usually even among those platform technologies, if you haven't tested coronavirus already in it, it still has to start at that early stage, at stage 1. And so that's kind of where we're at. If we had tested the SARS and the MERS virus vaccines and taken it to later stages, then we might have been able to pop in the COVID and jump a series of steps so that we can go to a later phase much earlier. That's kind of the point that we're trying to illustrate. So in this instance, we're going through these stages systematically, but as quickly as possible. So I think that, the most realistic thinking is that within a year's time, about this time next year, we might be in a late phase trial with a vaccine. But that's assuming that that vaccine succeeds at every stage, which means that it continues to prove to be safe, it continues to prove to have the immune responses that we need to have protection. All of these things need to be satisfied for it to continue to go through. That's why we'd like to have a number of different vaccines that are being evaluated all simultaneously. All of them, it's a big horse race all at the same time. It's not just one horse finishing. We need to have multiple horses finishing the race. And that means that we'll have a better supply of vaccines in general at the end of the year. Now, the next question would be, even if you have a vaccine or a set of vaccines that are licensed at this time next year, how are you going to deploy that vaccine to the population? And when I'm talking about the population, I'm thinking about the global population of 7.7 billion. And so that means that those manufacturers have to be able to make that level of vaccine and deploy it down to the person. So that is a logistics nightmare that we would have to overcome to get it down to these populations, rural populations, in other developing country settings, as well as here in our U.S. So again, it's going to be a continuing battle that you'll see that'll be around for the next 12 months to 2 or 3 years.
Okay.
Yeah, if I can just interject on a couple of things. One is what I've been trying to tell people about in terms of timing is right now, what we have is what you might call a behavioral vaccine. And by that, I mean, we don't have a biomedical vaccine. We have all the things you've been reading about in ways to keep yourself safe and protected from the virus, right? So social distancing, hand washing, cleaning surfaces, et cetera. The next step might be, but it's not really deployable for the entire global population, would be what we call passive vaccination or immunization. So mainly what we've been talking about are most people think of vaccines as active immune vaccines, and that means you get an injection, your immune system takes over, and then you're protected because you were injected with the vaccine. But there's also passive, meaning we give you somebody else's antibodies or synthetic antibodies, essentially, to give you short-term protection. So there's in the works right now, and some studies going on, what we call convalescent plasma. And that is where someone's sick with COVID-19, they recover, they now have very high antibody levels in their blood that protect them against future infection. So you can have them go to a blood bank, have their plasma taken off, and then do all of the safety checks on that you would for any other blood in the United States, and then give that back intravenously to someone else who may have been exposed or maybe already is sick or other ways. Now, obviously, that's not a way that you can get a vaccine to everyone in the US or everyone on the globe, but it might be a way for certain people. And then over time, as we've been saying, to get an active vaccine market.
Thank you. We are open to questions for the panelists. I have one question here from Lauren D. Giacomo. Do you anticipate COVID-19 mutating yearly like the influenza virus does?
There are a lot of things that we don't know the answers to yet. The good news about coronaviruses, and so far for this SARS-CoV-2 or COVID-19, is there's not been a lot of mutation. So we can't predict in the future what's going to happen, but the virologists tell us that as Dr. Chen said before, the fidelity is better for coronaviruses than it is for flu. And so chances of it mutating and being different and that we would need new vaccines all the time are lower for coronaviruses, but until time goes on, we won't know. There are these nice, there are over 2,000 completely sequenced SARS-CoV-2 viruses that are out in a database that people are looking at how much they've changed over time in all the countries where they've been. And there's not a huge amount there are some mutations, but not enough that it's escaping immune systems. So if it's already been circulating all this time and we're not seeing a lot of mutation, it's less likely that we're going to see escape, but we can't predict for sure in future seasons.
Right. Dr. Chen, anything to add to that answer?
I agree. I think that we really don't know. Based on the limited data that we have, we don't see a lot of mutations. So we're not expecting that it'll be acting like influenza does every year, where we have to catch up with the strain. But again, we could be proved wrong. So we'll see what the answer is next year.
So I remind you all, we're open to questions if you've got any additional questions. So while I'm waiting for another question, when I was in my clinical trial, all you did was draw blood from me over periods of time. You didn't expose me to the virus. So how do you know that I was protected?
So we have a number of ways that we can try to look at protection. So with the trials that you were in, and often our trials are to have two primary, what we call outcomes or endpoints. Those are safety. So like you said, looking at your blood for safety, but also making sure, asking about what side effects you might have. And then what we call immunogenicity. So for a lot, which means your immune systems respond. So for many of the viruses and bacteria that we are developing vaccines for, we have an idea about what we call a correlate of protection. So what is it in the immune system that correlates with or predicts protection? So in some cases, that's an antibody level. In other cases, it's an amount of cells or the way your cells react to certain things that you trigger them with. And if we can correlate those things, then we can predict pretty well whether or not you'd be protected. When you get to phase 3 studies, those are efficacy studies. That's where we vaccinate some people and don't vaccinate other people. We send them out into the world and they might be exposed to that disease. And then we see, are they protected? Those are really large studies and they're the last phase. But like if you were in, for example, in my study, Dr. Jarrell, was the H5N1, that virus never circulated in the United States, really. So all we could do was predict whether or not you'd be protected by seeing whether or not you had antibodies in your blood afterwards. We also, maybe Dr. Chen can explain our other, pretty unique to CVD, but some other places do it, these things called challenge studies, which are a really interesting way to see whether or not people are protected. Dr. Chen, do you want to say a little bit about challenge studies?
Sure, yeah. And some of the audience might have already heard this at different venues as well, but the CVD has actually been involved with developing human challenge studies for the past 40 years or more. And what we do is we take healthy volunteers, bring them into an inpatient unit, and actually have them get the infection. And in many instances, most of the time, what happens is that before that they come into the inpatient unit, they're vaccinated with either the vaccine or a placebo. They don't know if they got which one they got, so it's a randomized placebo-controlled trial. Then they come into the challenge unit, get exposed or infected basically with that infection. Let's say, for example, more recent one is cholera or influenza. And everyone has the same opportunity to get the infection. Then we observe them for a period of days afterwards to see actually who gets sick and who doesn't get sick. And then we can see, okay, well, some people had illness, some people didn't, but they all got exposed to the same virus or the same bacteria. Then we unblind ourselves, and with the data analysis with a statistician, we can basically see these are the vaccinees, these are the placebo recipients. The vaccinees only had this amount of disease, the placebos had this amount of disease, and now we can calculate the efficacy comparing those two. So it's a really unique way for us to be able to get at efficacy within a very short period of time, and actually the less number of people being involved in the study. Whereas phase 3 trials are usually many thousands of people, these challenge trials are about 100 people, or sometimes even less than 100, but it's on the order of less than 200. So again, time is money to manufacturers, but also less people being exposed to the actual infection and being able to license a vaccine that actually is effective is actually a great public health tool.
Yeah, you all are in a research university where we do a lot of clinical research. I would urge you to learn about it and participate in it. I participated in it for 2 reasons. One, I wanted to practice what I preach. That is, if I'm willing to enter a clinical trial, you should be willing to enter a trial. The other reason why I did it was because I wanted to experience firsthand what one of the best clinical trial places in the world is like, how it runs, how they take care of their subjects, and I have to give it a gold seal of approval. It was a terrific experience, so I would recommend it to all of you. We have a couple of other questions here. One from Melinda Hughes. How long does it take a vaccine to become available worldwide? You just told us about the problem. So in effect, how long does it take?
Jim, do you have an idea?
Yeah, well, it depends on a number of different factors. And if we're talking about, let's say, one of these coronavirus vaccines moves along through the phases, and then we find one or some number of them that are very safe and very efficacious, and now we want to have it available for the entire globe. What does that mean? Well, then the issue is what we call scale up. And that is, we can make a vaccine even at the CVD, or a small manufacturer can make it for hundreds or even thousands of people in a small place, in a lab, in a warehouse, wherever you're making. When you're talking about vaccinating the globe, depending on how many vaccines you need and how frequently, that takes a lot of manufacturers and a lot of manufacturing space. So it's a matter of, nobody is going to build all of that manufacturing space now when they don't have a vaccine and don't know which platform. Some of the platforms require cells, like cell culture. For a flu vaccine, it requires a certain type of egg, and many of the flu vaccines are made in eggs. So you have to get all of these eggs, inoculate them, take the virus out of the egg. There's a whole process that occurs at those manufacturing plants. So it will all depend on which rises to the top as the leading vaccines, as to how quickly and how broadly we'd be able to do it. No single manufacturer at this point, I would say, is going to be able to scale up to vaccinate the entire globe. So it would take multiple people using whatever platforms come along, or it might be multiple vaccines that work in different ways for different people. How long? It could happen over, let's say, 6 months to a year. But if we start seeing ones that are more and more likely, then the risk goes down and down in terms of the money that people have to put into it. So manufacturers would start making plants and redesigning their current plants to be able to manufacture this vaccine.
Okay, we have a question from Bonnie Bissonette on the microphone. Bonnie?
Oops.
Hey, Bonnie.
It's submitted.
Yep.
Can you hear me now?
Yeah. Yep.
Okay. So, do you think the emergence of shared research towards a vaccine for this pandemic might be transformative in terms of how scientific research and sharing across country borders might go on in the future? Or do you think this is a blip and we're going to go right back to the way it was before?
What do you think, Wilbur?
Okay, Bonnie, you always ask those tough questions. So I think that it's been very exciting to see kind of open data sharing and transparency at this time. This is the time when we need it. I don't know if it's going to be the way to go in the future, that people will continue to be willing to share like they are right now. Part of me thinks that when we start opening back up to normal business operations, that businesses again will make that financial decision. Sometimes they need to keep intellectual property secret and close to their chests. So that means that there will be limited data sharing, back to the kind of the old days. But I think that a lot of this has set a lot of precedents where we can try to be hopeful, at least as scientists, to continue to collaborate effectively, openly, internationally, and to see these kinds of things blossom more frequently so that we can be prepared for the next pandemic. So that's kind of my hope. But again, Bonnie, that was a difficult question. I think that, again, it's recognizing that there's a lot of openness now because of our attitudes in trying to react to a pandemic as a team, but then pessimism because, again, from the business sense, sometimes you do need to have trade secrets, and that means limited sharing.
Yeah, I think I'm optimistic, Bonnie and Wilbur. There have been some starts to this in the past that got pretty far along, but not as far along as they should. So many of you may have heard there's this whole coalition for epidemic preparedness and innovation that was funded quite well and was moving along after SARS and MERS, but now I think it's even more invigorated, and I do have high hopes now that people will realize that I think what's happened in the past is some people have thought all of these pandemic preparedness and people, they're wolf criers. It's you told us H7N9 was coming and you told us H5N1 was coming, and they fizzle. and now it's just going to be another fizzler. Well, now people realize that you're going to have these small, possible ones that maybe they never go as far, but some of them are going to break through like this, and we need to be prepared for the ones that do. And now I think people realize, no, we're not crying wolf. Like, we need to be better prepared, and we have the ability to be better prepared. We just all have to be in it together, and I think that is what's.
Thank you. We have another question from Mr. Joseph Frew. Is there a mic open, Alex?
Mr. Frew, your mic is open, if you can ask your question. He was referring to his question had to do with reports of South Koreans being tested positive for COVID-19 a second time. And what they worry is a reactivation of the virus. He asks, what does this mean for vaccine development?
Yeah, I'll start with that. And then Wilbur, if you have anything else to add. So our current knowledge, although for pretty much everything that has to do with COVID-19, our knowledge is limited. We're learning every day. But our current belief is that the vast majority of people who are infected develop high titer neutralizing antibodies, which means a lot of proteins in their blood that neutralize, meaning they attach onto the virus and prevent it from infecting at least cells. Now, can we say that that surely means that they can't be reinfected? Not yet, but it's good evidence that it's unlikely that at least right away that they can be reinfected. Most people believe that those stories are not that people are getting reinfected, but that they've had negative tests and then positive tests. And, it's possible it was either a poor swab or a false negative. We don't know for sure yet, but it doesn't look like now that people are likely to be unprotected early on after being, getting their first infection. Can we say that for sure yet? No, but I wouldn't necessarily read too much into those reports that you've heard about in South Korea and in some other places.
No, and I agree with that. I think that what I understood was that they did serial testing over days. And so on some of those tests, they were positive, then they were negative, and then positive. And, they weren't sure if those were reinfections. Now, to me, that means that the testing probably, we don't have a very sensitive test right now. So some of the times they're going to be negative, and you might have low amounts of virus and it just doesn't detect it. And one of the other ones, for whatever reason, becomes a little bit more positive, and so it comes up as a positive. I don't see that as reinfection. What I think would be more concerning is if you had a person who had COVID and then a month or two later, after their recovery, had a clear illness that, again, was shown to be COVID, then that would be a more clear case of reinfection. I think that what's being described in the South Korean publication and literature is a person who's on the mend but continues to have some positive lab tests later on as they're recovering. And that doesn't mean reinfection.
We have another question from, is it Aisha Mutani?
Aisha.
I don't hear anything.
Alex, are you able to see, did she write it? Did Aisha write? Question?
Try again, Aisha. Oh, she's dropped off.
Okay. I got two other questions. One from.
Able?
Oh, go ahead.
Hello.
Hello.
Hey, Dr. Campbell and everyone else. Thank you for being here. So my question, how long it takes? We know for drugs, usually it takes up to 12 years. And my question was just naive question about for vaccines, how long does it take? And if there is any emergency, would FDA approve, this time for those vaccinations for emergency like coronavirus? So can we develop vaccine in short time, very short time?
Yeah. Yeah, I mean, it's a good question. I think, the usual progress of vaccine development is on the order of, once it gets into phase 1 trials, if it continues on and makes it all the way to licensure, it's typically on the order of about a decade of testing. And a lot of that has to do with what we said early on, is you want to be very careful and safe. You also go kind of slowly through what we call the phase 2 trials, answering lots of different questions. That is, what's the right dose? Should we have an adjuvant? Should we have 2 doses or 3 doses? Do we need a booster? What about kids? What about older people? What about people with diabetes? All those questions get answered in those phase 2s. For these types of emergencies, all of that can be shortened somewhat as long as you're not really cutting corners. And the expectation is the fastest it could be would be about a year or a year and a half. And would the FDA make any special considerations? I think they would, but I think we all as Americans should also be proud of the fact that they're not going to license something if they don't, and that's what we're paying them for, feel that the data make us fully confident that it's safe and it's going to work. And it takes a fair amount of information for them to feel that way. So yes, we should pressure them to move. Us and they should move as quickly as possible, but we do not want them to say, okay, everyone in the United States should now get this vaccine, when we don't have high confidence that it is going to be completely safe. So again, I think it's 12 to 18 months is our best guess for as fast as we could have something.
So I have 2 last questions. One from Susan Buskirk about donating plasma and who might be a candidate for that. And the second from MJ Tui about a possible recurrence or resurgence of the virus in the fall.
I'll do the first one, Wilbur, you do the second one. So there is a program right now. Well, there are multiple programs in the US, but there's a program here at our institution. Anyone who has known to been infected here will be contacted to see whether or not they would be interested in donating plasma through our blood bank. So the director of our blood bank is in charge of that program, and she would be able to speak better to the details of it. But if you know someone or you are interested because you think you've recovered from it, then you can get in touch with Dr. Fontaine. So Magali Fontaine is the director of that program. And there are others in the state of Maryland and in other states. So if you have relatives elsewhere, they can look into that. I know in New York, the NYBC, if you just look at NYBC on Google it, they have programs all over the state there where they are collecting blood at their blood banks, and I'm sure in other places as well. So there are programs, and I think it's only for people who have recovered from disease for the plasma donation program.
Thank
Wilbur,
you.
do you want to take on the
The seasonality question?
Yeah.
So, for influenza, we know that when pandemics occur, they can occur in waves. Usually that second wave is about 6 months after the first. First wave is called the Herald wave. It's usually in spring. And then early autumn is when you see the second wave. Then you also have a third wave that comes on later. So for coronavirus, a lot of people are thinking that, yes, we might see a wave or a resurgence in the fall, as the question implied. So right now we're in spring. As it gets warmer, maybe the climate, the humidity changes so that these COVID infections actually die down a lot. But again, as the weather cools in the autumn time, and also when schools open up, presumably, that you have more people congregating into rooms, that you might have more opportunities for COVID resurgence in the fall. So that's a lot of the thinking, is that for pandemic preparedness for COVID, we're trying to be prepared for right now through summer, but then even if we see a dying down over the summer, that does not mean that we will not see. We're trying to prepare for the eventuality of a fall surge as well. So a lot of the planning that you see on campus or at medical systems around the state or even nationally are to continue to push for a surge capacity and capacity building in medical systems.
We had one last question, though, that I can't not ask. It relates to a topic that's been in the news the past couple of days, specific reasons why separate races might have a different susceptibility to COVID-19, and would that affect the way you might choose participants in a vaccine trial?
I can start with that. So the second part is, would that affect how we would choose people to be in the trial? No, all of our trials, we allow anyone who comes who is eligible to be in the trial. And that is, we want to have a good distribution. The distribution of the people in the trials, in our trials at least, typically reflect the distribution by gender, race, all of the demographics. The distribution within the city of Baltimore. And we think that that is important, that we have a good distribution and that everyone is included. Why might there be a difference in disease? We do not think there is any difference in susceptibility, meaning if you're exposed to the virus, no matter what your color, your race, your gender, your age, you have the same susceptibility to getting infected. The difference is how severe the disease is once you're infected. And unfortunately, it's been that people who are poor and people who may have already suffered from other what we call comorbidities, meaning like you come to the disease already with diabetes, heart disease, high blood pressure, things like that, that when you get the disease, you're more likely to have a severe outcome. So it's unlikely that it's actually the demographics that's leading to the problem, the more severe, higher severity. It's more likely that it's those people in those demographics have already underlying pre-existing conditions. And the other unfortunate thing is it's much harder to physically, socially distance yourself when you're someone without the economic means to do that. In other words, you're going to lose your job if you're someone who works day to day, hour to hour in someone's home, doing things that you don't have. Those people of that type, they can't sit on a Zoom or a WebEx like we are having teleconferences. They need to continue to make money the way that they're making money and to create, every day for their families. And so it's things like that also that I think, people should keep in mind. And it's been shown across our country and elsewhere that that leads to increased risk. So I think everyone should do that.
Dr. Chen, the last word.
So again, I think Jim said it. I don't think that there's a predilection for race or for gender per se by the virus, but I think that unfortunately there are disparities across the U.S. and those disparities lie in racial disparities, socioeconomic disparities, and that also leads to having higher rates of hypertension, diabetes, obesity, other things in certain racial categories. That again means that you have more severe disease, and that's an unfortunate disparity that we see. So I think our university has a center studying disparities, and so I think that perhaps those type of data will be studied and borne out after this virus is done. But I think that that's what's happening.
So I want to thank Dr. Chen and Dr. Campbell for a most enlightening discussion. Thank you for doing that. Thank you for giving us all that information. We look forward to hearing how all of this plays out, and I'll be in your first vaccine trial, I hope. In the meanwhile, these webcasts, we will have our next one next week. It'll be a virtual face-to-face. The topic that I would like to look at is the different activities in motion to try to control the pandemic in its current situation. Things like testing for COVID and other clinical trials of antivirals, et cetera. So until then, I look forward to seeing you next week. Again, to the panelists, thank you very much.